Everyone, myself and Vishnuvardhan from Department of Microbiology at Tirupattur Sacred College. Today I am going to explain about yeast, its industrial application and especially production of baker yeast using Saccharomyces cerevisiae, which is widely used in baking and fermentation industries. Introduction to yeast. Yeast are unicellular eukaryotic fungi. They do not contain chlorophyll, so they cannot do photosynthesis and depends on organic carbon source. They are commonly found on fruits, plant surface, soil, fermented food. Important point of yeast. Yeast are grow well in sugar-rich environment, which is why they are important in industrial fermentation. A common example is Saccharomyces cerevisiae. Morphology and cell structure of yeast. Yeast cells are oval or round in shape. Size range from 3 to 10 micrometer. Cell wall is made up of glucanase, mannose, cytine. Cell organelles include nucleus, mitochondria, vacuole. Reverse food material, glycogen, lipids. These reverse materials help yeast survive under unfavorable conditions. Reproduction in yeast. Yeast mainly reproduction by budding. Other methods include fission or rarely happen, sexual reproduction, formation of ascospores. Budding allows rapidly multiplication, which is very useful in industrial fermentation. In this image, budding in yeast is an asexual process in which is a small buds form on the parent cell, receive a nucleus, grow and separate to form a new cell. Growth requirement of yeast. Yeast needs carbon source, glucose, sucrase, maltose, nitrogen source, ammonium salt. Temperature 25 to 30. pH 4.5 to 6. Oxygen requirement needed for biomass production, not needed for alcohol production. This difference is the basis of industrial yeast production. Industrial importance of yeast. Yeast is industrially important because of fast growth rate, simple nutrition needed, high fermentation efficiency, genetic stability. Grace status, safe for conservation. Grace means yeast is safe to use in food and pharmaceutical industries. Role of yeast in baking industry. Yeast ferments sugars, tofu and produce carbon dioxide. Tofu rises. Ethanol evaporate during baking. Benefits. Increase tofu volume. Give soft crumb structure. Improve aroma and flavors. Increase shelf life. Yeast in brewing and alcohol production. Yeast convert sugar into ethanol, carbon dioxide. Type used. Top fermented yeast, ale. Bottom fermentation yeast, lager. Used in beer, wine, whiskey, bioethanol production. Different yeasts are selected depending on the product. Pharmaceutical and medical applications. Yeast is used to produce vitamin B1, B2, B6, B12, amino acid, and enzymes. Medical uses. Hepatitis B vaccine production used as a probiotic, act as a immune booster. Yeast in biotechnology and research. Yeast is a model organism in molecular biology applications, gene cloning, protein expressions. Produce recommendation product, insulin, growth hormones, also used in biofuel in research. Nutrition value of yeast. Protein content 45% to 55, rich in essential amino acid, vitamins, minerals. Advantage rapid biomass production, less land requirement. Used as a animal feed, poultry feed, nutritional supplement. Baker yeast characteristics. Commercially farmed Saccharomyces cerevisiae, non-pathogenic and safe, selected for high carbon dioxide production, low alcohol formation, widely used in bakery product. Baker yeast raw materials. Raw materials are used molasses, carbon sources, ammonium sulfate, nitrogen sources, phosphate, energy metabolism, magnesium, and trace element, water, air. Oxygen is essential because of Baker yeast produce aerophilic. Principle of Baker yeast production carried out under aerobic condition. Objectives: maximize yeast biomass, minimize algal production based on pasteurization effect. Pasteurization effect: presence of oxygen suppress algal formation and promote cell growth. Preparation of molasses medium. Molasses dilute to required sugar level. Classification removes of impurities. pH adjust to 4 to 5. Sterilization before fermentation. In this image, Baker's yeast is produced industrially by growing pure yeast culture in sterilized molasses through seeds and commercial fermentation, followed by separation and filtration. Inoculum development: pure culture maintained in lab, scale up process, flask to seed fermented, fermented to production fermented. Purpose: prevent contamination, ensure activity each cell. Aerobic fermentation: large fermented 10,000 to 1 lakh liter, continuous aeration and agitation. Control parameters: temperature 30, pH 5 to 4.5 to 5.5. Result: rapid heat multiplication. In this image, yeast produce more energy using aerobic respiration in presence of oxygen, but in absence of oxygen, fermented sugar to produce ethanol and carbon dioxide. Harvesting of yeast cell. Yeast separated by centrifugation. Concentrated yeast cream obtained. Washed to remove residual sugar. Processing and drying. Yeast pressed into cake or granules. Drying method: trump drying, fluidated bed drying. Purpose: reduce moisture, increase shelf life. Type of commercial Baker yeast: compressed yeast, active dry yeast, instant dry yeast. They differ in moisture content, storage stability, usage method. Conclusion: yeast are vital industry microorganisms. Saccharomyces cerevisiae dominate fermented industries. Baker yeast production is a controlled aerobic process, play a major role in food security and biotechnology. Thank you.